When considering the structure and chemistry of the amino acids, it's useful to begin with a free amino acid. And in this structure here, we see some interesting details immediately. First of all, that there's a central or alpha carbon that is decorated with 4 different substituent groups. There's the alpha amino group and the alpha carboxylic acid group. And these 2 groups will be the same for any free amino acid, as will the alpha proton. But what distinguishes the amino acids one from another is the identity of the R group. And we know that in nature, typically we encounter one of 20 different amino acid side chains. But before we get to that, let's consider too that those 4 different substituent groups makes an amino acid a chiral structure. The alpha carbon is a chiral carbon. The structure that's drawn here is the L enantiomer. And we will see that in nature we generally encounter, although there are exceptions to this rule, L amino acids. This L and D designation is commonly used when discussing amino acids and refers to the rotation of polarized light by glyceraldehyde. Also important to consider is the fact that the alpha amino group is a base and is protonated and ionized at physiological pH. So it's a base with a pKa of around 9. The alpha carboxylic acid group is also ionized at physiological pH as an organic acid with a pKa of around 2. What this means is that a free amino acid at pH 7 is a Zwitterion. Now, that's important to think about, but in the context of a protein, what we see is that the carboxylic acid group of amino acid 1 is condensed with the alpha amino group of amino acid 2 in a peptide bond. And so when we're considering the context of a protein, while of course the N or alpha amino group terminus of the chain, that is the alpha amino group of the first residue of the polypeptide chain, will be ionized, all the other amino groups are condensed in these peptide bonds. And so the N terminus is defined by that amino group, and the C terminus is defined by that free carboxylic acid group at the last amino acid of the polypeptide chain. As biochemists, we want to immediately interpret the structures of amino acids and proteins that we observe in terms of their chemical properties. So I want to practice that with the amino acid side chain arginine. And here you can see that I've omitted everything up to the alpha carbon. That is, I've eliminated the alpha amino group and the alpha proton and the alpha carboxylic acid, and we're just looking at the side chain. So let's think about this side chain. First of all, arginine is a basic side chain. It's ionizable. It's positively charged at physiological pH. And resonance among the 3 nitrogens in the side chain results in a relatively high pKa of above 12. Arginine is frequently involved in salt bridges that stabilize tertiary and quaternary structures of proteins. As well, those 3 nitrogens are engaged in extensive hydrogen bonding interactions in proteins. That functional group is referred to as the guanidinium functional group. And what we see is that at that first nitrogen that we encounter along the backbone of the chain, after that, at the zeta carbon, we're branched, and we have those 2 other amino groups. Of course, there's a 3-carbon aliphatic trunk to arginine as well. Since we've begun with an ionizable side chain, let's continue our exploration of amino acid chemistry by looking at more side chains which are ionizable. And again, the key here is that we're thinking of ways by which we can organize our understanding of the amino acids and their chemistries. So beginning with aspartic acid, and here I've drawn this as the conjugate base form, so aspartate, we see that there are some details that we need to learn about all the amino acids. First of all, they're 3-letter code, so asp for aspartic acid, which has a 1-letter code, D. Now, this is an ionizable side chain, as I mentioned, and we see that after its beta carbon, we have a carboxylic acid group with a pKa of around 3. I'm being intentionally vague about pKa's, and it'll be clear why in the end. But I think it's worth noting that we should be able to identify roughly what the pKa of an organic acid should be, and it's going to be somewhere between 2 and 4. But let's say, as we'll see, aspartic acid has a pKa of around 3. Stepping away very briefly from the ionizable side chains, the transamination of aspartic acid results in the formation of the amino acid side chain asparagine. ASN, ASN, 1 letter code N. Again, I want you to be paying attention to what sort of interactions these molecules might be able to engage in. Obviously, asparagine can engage in hydrogen bonding, both as a donor and as an acceptor. Back to ionizable side chains. Glutamic acid, here again I've drawn it as the conjugate base glutamate. GLU, GLU is a 3-letter code with E as the 1-letter code. Again, a side chain that is ionizable, carboxylic acid moiety with a pKa of around 3, and also has a transaminated version, glutamine, GLN-Q. The only difference between glutamic acid and aspartic acid is that extra carbon in the stock, if you will, of the acid. Moving on, here's arginine again, and I've given you its 3-letter code, ARG, or R as its 1-letter code, pKa of around 13. Here again, here's a better picture of what that sort of resonance structure might look like, wherein that positive charge is distributed amongst all of those nitrogens in the guanidino group. Another basic, that is to say positively charged amino acid side chain is lysine with a pKa of 10. 4-carbon aliphatic chain topped with a free amino group. Third basic side chain, histidine. And here I've drawn on the left the protonated form of histidine, positively charged at physiological pH with a pKa of around 7. It's always worth noting that histidine, with its relatively low pKa for a base, is subject to small changes in pH that will result in changes in the population of protonated and unprotonated states. And on the right here, I've drawn you the deprotonated version, the base form of histidine, without its lone pair on that deprotonated nitrogen. So those are our 5 amino acid side chains that we usually think of as ionizable, and that is that because the side chains are ionized at pH 7, physiological pH. Therefore, they're relevant for us when we think about the interactions that amino acids engage in. Again, aside from hydrogen bonding, what we're really talking about with ionizable side chains is their ability to engage in charge-charge interactions. In addition to the 2 acidic side chains and the 3 basic side chains, which again we consider to be ionized at physiological pH, I've included on this slide 4 other amino acids, which are polar side chains that we don't normally encounter as ionized side chains, but which are nevertheless ionizable. The first of these is cysteine, 3-letter code CYS, 1-letter code C, with a pKa of around 8. Again, we don't normally encounter cysteine in its conjugate base form, that is the ionized form. In fact, normally, and what's interesting about cysteine is that we encounter it frequently in extracellular proteins engaged in a disulfide bond involving 2 cysteine side chains. These disulfide bonds are frequently used to stabilize 3-dimensional structures of proteins. There's serine, 3-letter code SER, S-E-R, single-letter code S, with a very high pKa, greater than 13. Nevertheless, we encounter that serine oxygen acting as a nucleophile in the context of the active sites of, for example, serine proteases. So again, not ionized at physiological pH, but nevertheless, that ionization is important in various biological contexts. Threonine, another hydroxyl-containing side chain with a beta branching there, Again, not normally ionized at physiological pH, very high pKa for that hydroxyl group, obviously. Finally, however, we have tyrosine, 3-letter code TYR, single letter code Y. That hydroxyl also has a high pKa of around 10. Worth mentioning tyrosine, however, because this is an example of an amino acid which has some sort of confounding sets of characteristics. On the one hand, we have a hydroxyl group, which is polar and engages readily in hydrogen bonding. But we also have this large aromatic phenyl ring that obviously has very hydrophobic characteristics. And I want to emphasize with tyrosine that as we attempt to categorize for ourselves the amino acids based on those chemical characteristics, we need to realize that that any particular amino acid might have a disparate set of chemical characteristics that we need to keep in mind. Before we move on to consideration of other hydrophobic amino acids, I want to make a quick digression into histidine. Here I've drawn for you the tautomers of histidine, and on the far right we just see the formal labeling scheme for the imidazole side chain of histidine. Notice that the two nitrogens are labeled the delta and the epsilon nitrogens respectively. Now, in A, we see the conjugate acid form of histidine in which both nitrogens are protonated. But when we're drawing the base form of histidine, we might not necessarily pay too much attention to which nitrogen we protonate, and that might be perfectly reasonable, but it's worthwhile pointing out that those two are different structures. Let's look at let's look at the case of the free amino acid versus myoglobin, the relative proportions of the protonated states of those delta and epsilon nitrogens. If we look at free histidine in solution, we see that the epsilon form predominates, whereas in myoglobin, it's the delta nitrogen that's protonated. So it's important to note there's only that it's the structural details of a particular molecule, particular protein context, that determine the behavior of the side chain. And that's a theme that we're going to return to. So here is a cartoon of that myoglobin structure, and we see the proximal histidine, that is the histidine that's bound to the iron atom of the heme. And again, we point out that it's the delta nitrogen that's protonated, and in this case, engaged in a hydrogen bond with a nearby serine. And we see that the epsilon, again, is the nitrogen is bonded to that to the iron atom of the heme. So the structural context determines the details of the behavior of the amino acid side chain, is the point here with digression 1. I want to take another short digression and address this issue of why we're not memorizing pKa's to the 100th decimal place that we see, for example, in the many lists that are easily available on the web that give us not only the side chain pKa's of several of these amino acids, but also the alpha carboxylic acid and alpha amino group pKa's, which again we already know are not relevant for the most part in the context of a polypeptide chain. But let's look at a few more of those lists, especially paying attention to cysteine, which this list from Leninger says is 8.18 side chain pKa of cysteine, 8.33 here from this other website, and then a third 8.00. So we know the pKa of cysteine down to the 100th decimal place, and that would be 8.00. Now I'm emphasizing here only that it doesn't really matter what the definition of the pKa of the side chain is of the free amino acid. We will find that the context of the protein determines what the pKa actually is, and that's what's really interesting. So as long as we can identify a side chain as an acid or a base, and then think about whether that acid or base is ionized at physiological pH, we're ready to move on. But let's look briefly at the active site of the enzyme Hin egg white lysozyme. This active site is composed of two glutamic acid residues. Number 78 looks ionized. That makes sense to us given what we understand about the pKa of organic acids. But glue 172 is not ionized. It's the weak acid form still. And it turns out that having those two different protonated states for those two different glutamic acid residues is critically important to the mechanism of this particular enzyme. It also begs the question, which we won't go into in this lecture, of how you can achieve those varied pKa's in the context of an enzyme active site.